Welcome to Curiosity Uncovered. Today we're talking all things DNA, and we're even going to do an experiment about DNA. But first, we have to understand what DNA is. So, DNA is deoxyribonucleic acid, and it's found in almost all the cells in your body. And it is what we call a blueprint for life, or the blueprint of making you who you are. So, a little bit about the discovery of DNA. So we knew that Gregor Mendel did a lot of experiments using plants, and he was beginning to understand that traits were inherited from a biological mother and a biological father. Okay, and so fast forward a little bit, and then we find some material that was called chromatin in pus-filled bandages, and that was done microscopically. And then it wasn't until the 1950s that there was like a huge race in the scientific community to discover what the structure of DNA actually was. So enter a team from the Cavendish Laboratory and a team from King's College. So the Cavendish Laboratory team was Watson and Crick, and I know you guys have probably heard of those names before, James Watson and Francis Crick. And the other team was Maurice Wilkins and Rosalind Franklin. And probably Rosalind Franklin is the one that you might not have heard of before. And there's a reason for that. So Rosalind Franklin was doing X-ray crystallography. So we know a little bit about X-rays now, right? X-rays have a high form of energy. They can be disruptive to our cells. And this was in the early 1950s. So she was doing this work in a laboratory, and her laboratory partner was Maurice Wilkins. So she got a picture, the X-ray crystallography of what DNA looks like. And then Maurice Wilkins does a presentation, and they talk about some of the work that they've done. And James Watson sees that presentation. Well, he and Crick have been working on molecular modeling. So they have been like designing Tinker Toys to figure out what the shape of DNA is. They had already figured out a little bit about base pairing, which is the 4 bases of DNA: adenine, thymine, guanine, and cytosine. And they had already found from analysis that the number of A's and T's usually were very closely related, and the number of G's and C's were very closely related. So already we're seeing this picture of A pairs with T and C pairs with G. So then they saw this picture of X-ray crystallography, and they thought, oh, we understand. It's a double helix shape. Okay, so then they go in and they make these models, like actual, sort of like Tinker Toy models, and they start putting them together, and they understand that the A's and the T's fit together, the C's and the G's fit together, and they fit inside of the phospholipid sugar backbone. Okay, so this is how it works. So it looks like that ladder when you untwist it. It looks like this ladder, and the A's and the T's and the C's and G's are on the inside. So it was like this eureka moment. And this was over 70 years ago. And if we think about all the things that we talk about DNA now and how we're using DNA, that couldn't have happened without that discovery. Well, in 1962, Watson, Crick, and Wilkins were awarded the Nobel Prize in Physiology or Medicine for the discovery of the structure of DNA. Now, you may think, well, where was Rosalind Franklin in all of that? Well, number one, they don't award a Nobel Prize to more than 3 individuals in a category. And number two, she had passed away from cancer in 1958. And they don't award a Nobel Prize to someone posthumously. So there was no way that she was going to be a part of that Nobel Prize winning moment. And I think what happened is a lot of people forgot about her contribution to the structure, understanding the structure of DNA. So we didn't forget about it, okay? And I'm wearing my DNA earrings, if you can see them. All right, we sell these in our gift shop, and part of the proceeds of this particular brand of earrings goes to support girls and women in scientific careers. So we're still talking about your Rosalind Franklin. So what is the job of DNA, right? Because it has to have a job. So the job of DNA is to code for the productions of amino acids, which are then strung together in order to create proteins. So again, we go all the way back till we talk about DNA being a blueprint. Okay, everybody's DNA is unique, of course, except for identical twins. Okay, but everybody has their own unique DNA. 50% of your DNA comes from the biological mother, 50% of the DNA comes from the biological father. So that means that your body, everybody's DNA and everybody's body is a little bit different. So we're going to do a quick little experiment today on how to extract DNA from cells. And what we're going to be using is we're going to be using strawberries. So you can just take your strawberries, you can pick off your leaves, all right? And then you're going to put them into a Ziploc baggie. So one of the first things that we're going to need to do is we're going to need to, didn't take off my leaves, is we're going to break apart those cell walls because as we know, our DNA is located in the nucleus of our cell. Okay, so somehow we got to get into the nucleus of the cells to get this out. So what we're going to do is we're going to add water, a little bit of salt, and a little bit of dish soap. Now, importance for the dish soap because the dish soap is something that is going to break down that cell membrane, that phospholipid cell membrane, okay? So we're going to be able to put that in there, smush it up, okay? So we'll smush until we have kind of like a mushy mixture. And then we'll come over here and you can do something as simple as taking a paper towel and we pour the mixture through here. This is filtering, okay? So a coffee filter, something along those lines, so that what we're going to end up with then is just a pink strawberry colored liquid. Okay, then we're going to transfer our liquid into a test tube, or you can use some other type of container at home. So all of these things are materials that you can find at your house, and you guys can do this at home. Then we're going to add, and here's the tricky piece of this. It's got to be cold isopropyl alcohol. And this is 91% isopropyl alcohol. We buy it at Sam's because we use it a lot and we just store it in the freezer. That's all you got to do. And then once you add it, you let it sit there for a little while. And then you can start to see, I see a little strand of DNA right up here at the very, very top. Okay? But that's DNA. Right there. So that's how we know it exists. That's how we know it's inside of our cells as it comes up there. So if you have any questions, we're going to put a link on how to do the experiment so you can do it yourself at home with all the portions and the step-by-step instructions and a little bit better explanation of what's actually happening so that you can see you too can do genetics at home. And you too can be a DNA detective. Thanks for joining us today.